we're able to make very excellent aerosols in there. And what that means is you're able to bypass deposition in the mouth and the throat.
We're getting, you know, delivery as accurate as injections.
Essentially, we're trying to put a hydrophobic material like a phospholipid or a hydrophobic amino acid on the surface of that particle, create the right particle morphology to give you a highly respirable particle.
So your value proposition centers quite firmly around this idea that delivering drugs directly into the lungs is superior to local delivery in the mouth and the throat. Why?
In a mouth throat deposition, you can have local uh, tolerability issues. You can have cough. You can have candidiasis. You have a certain swallow dose of some drugs have an oral bioavailability, and you don't want that. You don't want GI effects. And so it's much more than saying, oh, yeah, the engineered particles are 3 times as efficient. You have all these follow-on effects. And you can also get access to the deep lung with these formulations. So essentially, the inventive step is we're able to make very excellent aerosols in there. And what that means is you're able to bypass deposition in the mouth and the throat. So essentially, our head is a filter. So we've evolved over dusty conditions and campfires and things like that. And so by design, we try to keep particles out of our lungs. Particle engineers are trying to bypass our natural defense, get them deep into the lungs, target, and targeting is a key word there. So you're getting high concentrations of drug where they need to be.
And how did you come up with this solution?
It's by necessity. You're trying to deliver high doses to the deep into the lungs.
Right, but I guess where did this idea actually come from? Because you're saying that taking a blowing agent and blowing holes into nanoparticles, kind of like creating wiffle balls, and that produces a better delivery profile. Is that right?
Maybe it's biomimicry of sorts. There's a whole field called aerobiology,
Mm-hmm.
how fungal spores and pollen and things like that move.
Yeah.
Nature's particles are low density particles, and so that served as some of the inspiration for that as well.
Okay, and then like the idea of basically adding this agent that explodes the particle, where did that come from? And is there precedent for that?
It
Or
actually doesn't explode. That's
okay.
that's
Well then maybe can you take me through the process of actually creating the spray?
To
well,
create, go ahead, John.
no. I was just going to say it actually kind of originated through several people. We didn't make wiffle balls at the first. We made grapes, dried up, wrinkled grapes, and lots of other things in between. So it's a nuanced story. But Jeff Weirs, Dan's co-worker, who's giving the keynote speech at the respiratory drug delivery meeting next week down in Phoenix, he was the one that came up with the blowing agent. But it turns out you can also make wiffle balls with solvents, and but with other solvents or other liquids than Jeff used. But the wiffle ball, as Dan said, is nature invented it a couple billion years ago in fungal spores, and we went back in the literature and dug up some EMs, and there's a particle that looks exactly like these wiffle balls that you're talking about.
Yeah, okay, well, super helpful. Can you kind of just describe the manufacturing approach, kind of like the starting material and the excipients and what's the process of actually creating these unique sphere structures?
Comes out of actually Alliance Pharmaceuticals back in the late 90s. And Jeff and Tom Terrar were working on blood substitutes, and they're working with perfluorinated materials. And so Jeff has a very colloidal view of the world. And so it's a natural, I
Well,
would say
that
it's a
is
natural progression, but it was a significant innovation for sure.
Okay, all right, so how are they made?
So the heart of the technology is an oil and water emulsion. And the oil is it's called perfluorooctylbromide. So it's a chain of carbons with it's perfluorinated, so it's all fluorines, and the tail of it has a bromine on it. So a C8 chain. And so it's been described as liquid Teflon. And so if you ever seen the movie The Abyss, where they put the little mouse in the liquid, that is a liquid breathing medium, and it was probably supposed to be perfluorooctylbromide. So it has a high oxygen carrying capacity. We're not using that property here. We're using it as a material to form a it's not a blowing agent, but it's a pore forming agent. So a way to think about it is you make an oil and water emulsion, and when you spray dry that, you atomize that emulsion. So you have an atomized droplet with thousands of emulsion droplets in it, right? And those emulsion droplets are submicron. And as you evaporate that atomized droplet, the emulsion droplets that don't really diffuse all that quickly, so they get scooped up on the surface of the atomized droplet as they dry. So I left up something out. The water evaporates first. The water is more volatile, right? The blowing agent has a boiling point of 140 C. Water evaporates first. In a second stage, the blowing agent evaporates. As John intimated, it's a bit of a misnomer to say that it explodes the particles or it's a blowing agent, because rather I view it as more of a templating agent. So it occupies space as that atomized droplet recedes, and in the second stage of drying, that oil evaporates.
Wow, interesting.
Leaves behind a sponge structure, so it doesn't violently, boil off like that.
Right, okay.
And then in that technology, you can think, oh, I have an emulsion. And here's the other piece of this is that most people out there who are spray drying are just dissolving everything in one, either water or in a co-solvent, something like that, single phase solution. There's lots of advantages to moving to an emulsion, right? And the way I kind of look at it, if you look at the oral drug delivery world, they have hundreds of excipients. Really, it's such a rich world in terms of, as a formulator, all these things in your toolbox. Whereas in the inhaled world, because there's so much concern around what we're putting into the lungs, we only have, like, in DPIs, probably 10 excipients that are in approved products on that order. And because of that, it sort of has driven a lot of the creativity in terms of engineered particles, because you don't have this toolbox of excipients. So you have to think about ways to combine the excipients you have in different ways, and emulsions provide you an additional dimension that really helps. So you can dissolve the drug in the continuous phase in the water.
Mm-hmm.
You can suspend the drug in the feedstock. So you have two suspended species. You have an emulsion droplet. You have a drug particle. So it gives you a lot of creativity in terms of feedstock design.
Is perfluorooctylbromide, if I'm pronouncing that correctly, one of the approved excipients or one of the tox liabilities of that compound?
It's used in Toby Podhaler.
Okay.
It's used in an approved product. We've never called it an excipient. We call it a processing aid, because it is the amount in Toby Podhaler is less than 0.1%. So we wouldn't call water an excipient, right? So water is a processing aid and PFOB is a processing aid. So we've qualified that toxicologically.
So if we already kind of validated this with the Toby Podhaler, why didn't this become kind of standard of innovation for all DPI products?
Yeah, we've been screaming it from the mountaintop. It's just a slow process, and I would say maybe the shortest answer is entrenched players dilemma. You have a lot of people into lactose blends and very successful products out there, and you're constantly kind of fighting against that. It takes small companies that are not invested in this to actually take that risk usually. And I went through this at Novartis because they have a lactose blend a whole group that does that, and we were, let's say, constantly trying to prove that engineered particles, the advantages of engineered particles, even after we got acquired. 10 years into the acquisition. Um, yeah.
And then I guess for different molecules or different combinations of molecules, I'm guessing the performance characteristics almost across the board is, like for a list of questions that you might ask about DPI, delivery is going to be very, very dependent on the molecule, the molecules, the ratio. Like, how do you even think about that? Like, what do you tinker with to optimize the product? Is it mostly like, oh, I've got this vehicle carrier, the wiffle balls, and then really what you're trying to tweak is like, the relative concentration of the drug, or are there other parameters as well, like in the manufacturing that really impacts the delivery profile, apart from the specific compounds and doses and ratios?
Um, I'd start with every molecule has what I said before, like its own personality. So there's always a twist with every molecule. Um, in really broad brushstrokes, making the particle engineering piece is actually straightforward, and we understand that quite well. Essentially, we're trying to put a hydrophobic material like a phospholipid or a hydrophobic amino acid on the surface of that particle, create the right particle morphology to give you a highly respirable particle. Usually that's pretty straightforward to do to get the requisite aerosol performance. The other brushstroke is achieving the right chemical stability of the drug, and that's going to be necessarily drug dependent. And it's understanding the degradation pathways of the drug, and controlling them. So, and unfortunately, when you look in the literature, there's all these groups out there that are, that have a spray dryer and they're spray drying particles, and they're making good aerosols, reasonable aerosols, but they can't. They don't really show a lot of stability data, which is ironic because that's actually the harder problem. The other piece that's not appreciated is flow rate independent delivery.
Yeah.
So you might be able to get a high delivery efficiency, but if your delivery efficiency is highly flow rate dependent, that doesn't help you, right? So patients have different mouth throat geometries, and they have different breathing profiles, and you want to make sure that, let's say, 90-95% of their patients get their dose. And a lot of that's ignored in the literature. And here's what's painful. I was at a conference a couple of weeks ago in Dublin, and some people the calculations are wrong. How they demonstrate flow rate dependence, it's wrong. So anyways, that's my soapbox there.
So can you look at a molecule and kind of like predict how easy it's going to be, ultimately in this process, just look at the molecule and just sense the parameters you need to work with, or is it something very empiric where you have to go through a particular process of optimization?
No, it's not empiric at all. Actually, we look at all the properties, octanol water, partition coefficient, solubility, crystal forms, dose is huge. That can exclude
Yeah.
a certain device and so forth. And the guiding principle, I think, for guys like Dan and I and our colleagues is we want these formulations to not have any extra stuff. You want to put the smallest amount of material in the particle. No lactose blends where you're just having people, inhale large amounts of lactose. So but it's not empirical. You can. And we're sort of technology agnostic. We look at the property of the drug, the properties, and that dictates for us all kinds of things when we go to make a commercial
Okay.
product.
How do you even figure out the right dose for an inhaled therapy, especially if there's like validation from an oral systemic approach? How do you actually know, what to do there?
You don't. It's a good start that you know what the oral is, but the gastrointestinal processing, what goes on down there in the GI tract is awesome, awesomely complex, and a lot of drugs are purposely meant to be taken into the GI tract with as high a bioavailability as possible. We're different in the sense that small molecules, by and large, they shoot through the lungs like a rocket. So often we want them to cool their jets and stay around a little bit and have some efficacy. So it's a fun game when you've been in it for a while because there's so many variables. But once you've played with those variables for decades, like we have, it starts to be easier.
Okay, so I have a bunch of questions that ChatGPT has guided me to ask, which I find super helpful for a neophyte to both ask and learn as we go. So the first one is on emitted dosing. Does the dose actually leave the inhaler? And how do you quantify that or characterize that?
Yeah, there's several definitions of dose, but the emitted dose is how much you essentially you actuate the inhaler and you capture the material on a filter.
Yep.
And you can either gravimetrically measure that or use HPLC to assay it.
Got it. Got it. So I guess like what percent of the product is emitted versus left behind in the chamber?
90%,
Okay.
90%.
And that's consistent delivery, dose, uniformity, each dose, each use gives the same dose. What might impact that uniformity, whether it's for other products or this one in particular? What would you need to test and evaluate?
That depends a lot on the device.
Okay.
We can make liquid devices, vibrating mesh devices with extremely high efficiency and low variability with controlled breathing. And then we have old style nebulizers that are just blowing out drug all the time, and you're kind of pulling breaths off of that. So it's what's great about these new engineered powders is that the efficiency has really gone up, and with that the variability's gone down.
Okay.
So we're getting delivery as accurate as injections.
Let's talk about aerodynamic particle size distribution. Maybe you can characterize the particle size, the median mean, and kind of distribution of the particle size and how reproducible that is, and how do you actually quantify that?
Yeah, it's it's a tool called a cascade impactor. So the most common one's an NGI, next generation impactor. So essentially, it inertially separates particles, right? So it operates in there's different stages in it. So you think of it as you have each stage has a certain number of jets on it, right? So the first stage has, I think it's got like 6 holes in there. They look like they're about 1/4 inch, right? The next stage has more holes, but they're smaller, and you get progressively finer as you get into that impactor. And so you can run an aerosol through there at a prescribed flow rate, flow volume, and the big particles will go in the first stages, and the finer particles will go in the latter stages, and you can generate a distribution from that.
Okay.
The key parameter there is that it's inertial impaction. So each stage represents what's called a D squared Q cutoff. D is the diameter of the particle, and Q is the flow rate.
Okay, got it. What is the median flying size?
What is the median?
Like the MMAD, so the mass median aerodynamic diameter. I'm just trying to understand the distribution of particle size with the approach.
So it's basically finding so you have you run it through the impactor, you look at the masses on each stage, you have a distribution, and then the median of that distribution is the MM.
Yeah, no, I understand what it is. I'm asking what your results show.
Oh, you're getting MMADs in 2.5.
Okay. Is the aerodynamic size essentially really all that matters for the distribution? Like, oh, if you're in that 1 to 5 range, you're likely going to be distributed reasonably uniformly, across the lungs. But I guess in your case, because you have this tweak, right, to the structure, does that kind of change the way that we think about like particle size, the ideal size? Does it give you a greater range of sizes?
So, okay, let's go back to an impactor works by D squared Q, right? So it's the product of those two. And so you can have you'll read a lot of places, even reviews of the last few years, 5 microns and below are where what we want to target for a, respirable particle. Well, you can have a 10 micron particle if the Q is low enough, that's respirable as well. So you have these sort of statements that have proliferated throughout the literature that aren't true. And so we're trying to sort of change the world and get people to think more about D squared Q, because that's how an impactor works. And that's how what dictates throat deposition.
Right.
But as a very crude estimate, if you saw something that's less than 5 microns, you're like, yeah, that's probably where we want to be in terms of an aerosol. But if you inhale that at a high enough Q, you can make it a bad aerosol. So
Okay. What percent of the drug substance then falls into that aerosol range? Whether it's like 5 microns or less, or is there some additional leeway given the structure of the particle?
that's typically what when people say delivery efficiency, which is
Yeah.
which is a very catch-all term,
And that's what you're referring to.
you're talking about it's really a fine particle fraction,
Yeah.
and people define it different ways. Some people, the Europeans, will say FPF less than 5 microns,
Fine
which
particle
again
fraction.
is a size-centric approach, which is not necessarily the language we should be using. The challenge is that size is very we all understand that. Most of us don't think in D squared Q, right?
Right.
Very
Right.
abstract concept.
But I guess with your platform, it brings the importance to light quite dramatically, right? Because you have two particles of the same size. One with the wiffle ball structure is going to be very different to one of the same size without that structure. And the only way you capture that is with the Q, I guess, right? Am I understanding that correctly?
Yeah, well, the wiffle ball has a lower aerodynamic size owing to its density, um, right?
Right, right, okay.
Geometrically, they're the same, right? They have the same diameter geometric wiffle ball and a
Right.
baseball.
Yeah.
Aerodynamically,
Mm-hmm.
the aerodynamic diameter is the geometric times the square root of the density, right? So
Right.
the density of a wiffle ball is much lower than that of the
Right, 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 exactly. Okay. And that's the Q.
Q comes into the flow rate at which you
But
What is the Q referring to then, sorry?
I'm sorry.
flow
I'm s-
volumetric flow rate of inhalation.
And what is that? Can you define that?
Yeah, so we have a passive device, right? So if you have a
Yeah.
passive DPI, put that in your mouth and you inhale through it, right? There's a certain flow rate that's pulled through that device, and it's a function of the device resistance and the patient's inspiratory effort, right? So there's a volumetric flow rate, meters cubed per second.
Right,
I,
okay.
I wanted to interject
Sure.
another
Please.
great quality of these particles that we're making. Powders are usually associated with cough. People think, oh, yeah, dust. That's you make a powder, you're gonna make
Yeah.
people cough, and that's been true with a lot of inhaled powders in the past and some of the early stuff. But the way Dan is making them right now, making our eSpraye powders, there's no cough,
Yeah.
and
Yeah. And you mentioned that.
that is a that's an enormous breakthrough.
I get
So
Right.
that. Yep.
this is like a grand slam with powders that
Okay.
they have this quality.
Yeah. Yeah. Okay. Very helpful. Maybe just coming back to density, right? Because I guess that's the point you're making, that what differentiates the spray from other approaches is that it's lower density because it's porous. So how do you account for that? in these measurements of the fine particle characterization? Because like we're, if I'm understanding correctly, we're talking about particle size like certain microns, but the micron size doesn't adjust for density, right? And that's like when I was talking about Q, that's why I mistook Q for density. Because I'm thinking like, oh, okay, if we want to adjust the right particle size for density, we need to multiply or divide, right? Adjust for density, measure again, because like you're saying, maybe you have a 10 micron particle that actually acts like a 5 micron particle because it's less dense. So how do you actually quantify that or integrate that into the calculation and like interpreting the, I guess, like fine particle analysis, like the MMAD?
That's inherently done by the cascade impactor measurement. Let's go back to when we spray dry powders.
Right. So the end result is the MMAD that's influenced by the particle size, but also the particle density. Is that the message here?
Yeah,
Okay.
and the flow rate. So when we spray dry a powder, we out of the spray dryer we have bulk powder, right? It's not in a device, it's not in a capsule or anything. We characterize the size of that using laser diffraction. So we know the geometric size distribution of those particles, right? That says nothing about their density. In order to answer the density and question, the cascade impactor pulls all of those effects into one. It accounts for size, density, and flow rate, and it separates them inertially by d squared q.
Right. Okay. Super, super. Thank you. I am slightly less amateur now than I was 40 minutes ago, so thank you so much.
Every day is a school day for me.
And so you'd mentioned, so like flow rate dependence is a key differentiator, and you're testing across a variety of flow rates. So what kind of variability do you see across flow rates?
So Jeff Weirs has come up with an index for that called the Q index, where Q is flow rate. And like a high flow rate dependence would be a Q index of essentially that it's a slope of the delivery efficiency versus the pressure drop across that inhaler, which is flow rate, right? Yep. And so things with a high slope have a high flow rate dependence. You want a very flat slope. You look at like lactose blends, they might be in the 40s. So highly flow rate dependent. You look at engineered particles, we're able to get things below 5 Q index. So essentially, flow rate independent delivery. I mean, we have a paper from about 10 years back for Toby Podhaler, and we took all these breathing profiles of cystic fibrosis patients. So really huge variability in how people breathe, and also their in terms of their inhaled volume, their maximum flow rate, their ramp rate, and we use those to we recorded those breathing profiles and then use those to aerosolize data, aerosolize powder, and run it through an NGI. And we had 61% delivery efficiency across a huge variety of patients. And it was a really astounding result. I mean, it was absolutely remarkable, showing that all those patients would get their dose.
Interesting.
And that's not common. There's a lot of products out there have a high flow rate dependence.
Mm-hmm.
I think the other thing about what we're doing is combining multiple drugs in one particle. Talk about the advantages of that compared to the blends.
Yeah, that was going to be my next set of questions.
Oh, good.
So thank you.
Yeah. Thanks for the prompt, John. So and I'm fresh off my talk in Dublin, so this is actually a good timing for this. One of the challenges, if you look at the asthma space, and actually a lot of spaces, they're going to fixed dose combinations, right? And if you have a lactose blend, you have let's say a 50 micron lactose particle, and the lactose is just a carrier. And then what you do is you take a drug and you micronize it, you blend it with that carrier. And so you have this lactose boulder with all these little drug particles on it. And so the patient's inspiratory effort has to detach that drug from the lactose. And the reason I'm talking about lactose is it explains how engineered particles are superior. So a patient is asked to inhale forcefully in order to get that drug off the lactose. It's very cohesive. So when you go to a fixed dose combination, now you have a second drug in there interacting with the lactose, the second drug interacting with the first drug, and being able to get those both off of that lactose carrier at the same delivery efficiency is very difficult. And it's been termed, and this term is by people who work in lactose blends. It's not by a critic of them. Is multi-particulate nightmares. Understanding how to handle these multiple interactions between these particles. And so with an engineered particle approach, with two drugs like for ensifentrine and nintedanib, we can put both drugs in the same particle, right? So they're co-located, and so every particle has the same composition. They're both co-delivered to the lungs, and you don't have any of this multi-particulate nightmare issue.
How do you figure out the appropriate ratio? So like now you have the challenge of identifying the appropriate dose, not just for one particle, but for two, right? For one drug, but for two.
Yeah, dose is a challenge. So I mean, for this program, for ensifentrine and nintedanib, you actually have the luxury of you have monotherapy development programs, and you can look at what their dosing is, and you have to do some math to figure out what do I think their lung dose is, because that's what's important, is what that lung dose is. And again, it's a fuzzy number, but you have to start somewhere. So you get an idea of the lung dose of this monotherapy, and then you have an idea of what the ratio of those needs to be. I've been in other situations back at Novartis where you'd ask a medicinal chemist and biologist, what's the dose? And they'll give you 100-fold range. And as a formulator, you have to deliver. So when the range is like 2 or 3-fold, that's very straightforward to formulate around.
For uniformity, I guess like the images that I've seen, there were some beautiful, perfect wiffle balls, and there were some like, not so perfect wiffle balls. How do we think about uniformity and tolerance when it comes to just like deviating a little bit from that pure wiffle ball structure? I guess it kind of just gets down again to the density measure and homogeneity of density when it comes to that.
I would say it doesn't matter. I mean, in terms of
Yeah.
looks don't matter when it comes to particles. I think,
Okay.
yeah, the SDMs that you'll see in papers published, they're always sort of the poster child, really the most beautiful particle you've ever seen. But in a milligram of powder, there are, I don't know, billions of particles. And so you're always going to find some that are ugly.
Okay.
It's all about performance in the end. It's not about
Mass performance, yeah.
Got it, got it. I'm not sure if this is a particularly relevant question for this context, but X-ray powder diffraction, is the drug crystalline or amorphous, or is that just not the right question to ask in this context? Because these are wiffle balls. I'm not sure if you crystallize a wiffle ball.
Yeah, it's a very good question. It's so because in engineered particles you can introduce the drug in many ways into a feedstock. You can dissolve it, you can suspend it. You're trying to always you're trying to control the solid state morphic form of the drug.
Yeah.
And ideally you'd be at either extreme. You'd either have everything dissolved and you're when you spray dry a particle, you have about 100 milliseconds or so between an atomized droplet and a particle. It's so fast that most drugs will not crystallize. If they're dissolved, they won't crystallize over that time period. And because of that, they'll be amorphous in the particle, and you use X-ray powder diffraction to confirm that they're indeed amorphous. If you go to the other extreme, if you have a suspended drug particle, as John was talking about before, quite often, as you're saying, these drugs can shoot through the lungs. And if you want to slow them down, there's some benefits to keeping them crystalline. They'll have a lower solubility, a lower dissolution rate. And if you have a crystalline drug in the particle, you want to confirm that it's indeed crystalline by X-ray diffraction, and it's the same polymorph that you intend to develop. You don't have any polymorphic changes or anything like that.